introduce Mahwash Jamy. So Mahwash completed her undergraduate studies in Australia before obtaining a master's degree in evolutionary biology through the Erasmus program, during which she did several research projects in Sweden, in Germany, and in France. And she's now doing her PhD in Fabian Berke's lab at Uppsala University in Sweden, where she's been interested in pinpointing enigmatic Protist lineages at large. And today she'll talk to us about the use of long metabarcoding to investigate the phylogeny and taxonomy of eukaryotic environmental diversity.
Hey, thanks a lot, Laura, for that introduction. And thank you to the organizers for organizing this meeting and for having me. So today, as Laura mentioned, I will be presenting a method that we developed for long-read environmental sequencing. And I'll talk about how we can use it to study environmental diversity in a phylogenetic perspective. And then I'll finish by very briefly talking about some of our ongoing work. Sorry, okay. So let's start with the standard metabarcoding approach. So for eukaryotes, people typically collect their environmental sample and then amplify a barcode. Now this barcode varies on the group of organisms that you're interested in. For instance, if you're studying fungi, people will amplify the ITS markers. For studying protists or for microbial eukaryotes, the most commonly used barcodes are the V4 or the V9 regions of the eukaryotic 18S gene. So these are fragments that are less than 500 base pairs long, and they can be amplified and then sequenced with Illumina. So this generates millions and millions of reads, and it's a really powerful way of identifying organisms present in your sample. And this is done by doing pairwise similarity searches against reference databases. Now, over the past decade or so, there have been many metabarcoding surveys that have been carried out. Some on a truly global scale, like the Taro Oceans Project and the Malaspina expedition. And these studies have generated really large sequence data sets of the V4 or the V9 regions. And these have allowed us to do things like studying Protestant communities in really diverse environments. We've also discovered lots of novel diversity. And this data has also allowed us to look at patterns of diversity at different scales, local and global. However, this metabarcoding approach has one big limitation. These short reads contain very little phylogenetic signal. And this makes it really challenging to answer ecological and evolutionary questions that rely on phylogenetic inferences. So, for instance, it's hard to know where a new lineage belongs in the tree of life because it's so challenging to build phylogenies. And there are still many unanswered questions about the role of evolutionary processes in the patterns of diversity that we see. So there is clearly a need for longer environmental sequences. And it's a need that cannot just be fulfilled by Sanger sequencing because it is expensive, it's low throughput, and it is labor intensive. So this brings me to our project. And our aim here was to develop a method to do environmental sequencing with PacBio. And we wanted to sequence not just the whole 18S, but almost the entire ribosomal DNA operon. We then wanted to use the increased phylogenetic signal of these long reads to firstly assign taxonomy to the environmental sequences, and then to construct phylogenies of this environmental diversity across broad taxonomic scales. And this is facilitated by the fact that the 18S and 28S genes have conserved and variable regions. And so that allows you to build phylogenies at different depths. So for our pilot study, we obtained 3 soil samples, the details of which are not so relevant here. But we used general eukaryotic primers to amplify around a 4,500 base pair fragment of the ribosomal DNA operon. And then we sequence this on 3 SMART cells on SQL on PacBio using circular consensus sequencing. So PacBio has this reputation of having very high error rates, and it is true. So the raw error rates are around 14 to 15%, but this gets lowered significantly by circular consensus sequencing. And the way this works is, so you have your DNA amplicon over here, and this gets circularized by these hairpin adapters. This now means that the polymerase can go round and round and sequence the molecule many times. And each turn around the molecule is called a pass. So while the error rate is high, these errors are randomly distributed along the length of the pass. And so once you generate your consensus sequence, circular consensus sequence, or CCS, it has a much lower error rate. And in general, this means that the more passes we have, the lower the error rate. So from these 3 soil samples, we generated 113,000 CCS. And to curate these, we developed a stringent filtering pipeline that further filtered based on quality. It removed artifacts, and we also removed chimeras. But importantly, at the end of all of our stringent filtering, we extracted just the 18S and the 28S genes from the clean reads. So we didn't extract the ITS region because it's too variable across all of eukaryotes. And we then clustered these sequences into OTUs based on 97% similarity based on the 18S gene. And then after discarding singletons, we were left with 650 OTUs, or operational taxonomic units. These 650 OTUs were then treated in a phylogenetic framework. And the first thing we did was to assign taxonomic labels to these OTUs. For this, we used only the 18S part of the gene, and this is because the reference databases are so much more comprehensive for 18S than they are for the 28S gene. So to do this, we start by generating an 18S tree. And this is a tree that contains both our OTUs and also reference sequences that are available. So in this case, we derived our references from the Silva database. We then prune away the OTUs, leaving behind an 18S tree that's composed of references only. And the rationale for this is that we want to build a reference 18S tree, but one that's also using the information in the environmental sequences. So now that we have our reference tree, we use the labels at the tips to annotate the inner nodes. And then we can place the OTUs one by one on each of the branches of the tree using phylogenetic placement. And then for each placement, we calculate the probability that the query belongs there. So for instance, what is the probability that this OTU sequence belongs to this branches on this over here, or over here, or over here, etc. And here this is represented by the by the size of the circles. So we then use all of this placement information to calculate the taxonomy of the OTU. And in this toy example, this particular OTU sequence, we can be 100% confident that it is an alveolate. We can be perhaps 95% confident that it is a ciliate, and about 60% confident that it is ciliate genus A. The result of this is a table where for every OTU, we have the taxonomy to the appropriate taxonomic rank, and this is followed by the confidence level for each taxonomic rank. Secondly, the sequences are annotated to the appropriate taxonomic rank. So in the image to the right, we can see that this OTU branches very closely to the references. And so it's annotated at the genus level. Here it's labeled as Pomilla. This OTU in the figure on the right branches deeper, and so it's annotated to a higher rank. And this means that we don't really need arbitrary similarity thresholds for deciding what level we should annotate to. So in this way, we're able to label the 18S part of the OTU. And because it's physically linked to the 28S molecule, we can transfer the annotation to the 28S gene as well. And finally, so once we had labeled our OTUs, we used both the 18S and the 28S genes to construct phylogenies to investigate the diversity of our soil samples. And this resulted in this global eukaryotic tree. So the branches in orange represent our 650 OTUs, and the branches in black represent reference sequences. And these are reference sequences for which the 18S and 28S were derived from the same species. And then the purple dots show bootstrap values above 70%. So doing this allows us to visualize the diversity present in an environment. In other words, it allows us to phylogenetically resolve an environment for the first time. So there are a few things to note about this. The first is that this is a relatively well-resolved phylogeny, and it spans all of the eukaryotic supergroups. The second thing to note is that some clades are almost completely orange, which means they're almost only composed of our environmental sequences. So this is particularly true for example, the Rhizaria over here. We can compare this to the Metazoa, which contain a lot more black branches or a lot more references. And this just goes to show how biased and how poor our databases are, especially the 28S database. It also shows how powerful our method can be for improving our databases. And then lastly, our method allows us to investigate the relationships between the environmental sequences themselves. And we can do this in a much more robust and a more powerful way than the standard metabarcoding approach. And this is of course facilitated by the fact that our sequences are taxonomically labeled. So to summarize so far, I have shown you that we can sequence environmental diversity with PacBio, and we can analyze it in a phylogenetic framework. And this is work that was based on 3 soil samples. So now I'll change gears slightly and I'll show you some of the work that we are currently working on. So for the study I just talked about, we had used 3 soil samples from Tibet and from the UK. But to generate a dense and a comprehensive 18S 28S dataset that can then be useful for investigating various evolutionary questions, we needed more than just 3 soil samples. So we got a few samples from several different environments. So we got some freshwater samples, and these are represented by some Swedish lakes, as well as a Canadian permafrost thaw pond sample. We got more soil samples. 2 were from peat, 1 from a boreal forest, and 1 from a tropical forest. And then we also got some marine epipelagic samples, 1 from the west coast and of Sweden. And we also got several Malaspina samples corresponding to the surface layers and the DCM layers. And then lastly, we got some deep marine samples. So these were the corresponding mesopelagic and bathypelagic samples from the Malaspina, and 1 sample from the Mariana Trench. And then for all of these samples, we did the same thing as before, and we amplified ribosomal DNA operon. And this time we sequenced the amplicons on the new PacBio Sequel 2 on 4 smart cells. And we obtained a staggering 12 million raw CCS reads. So this is like 100 times more high throughput than the previous sequel 1. These 12 million CCS were then filtered and clustered into more than 18,000 OTUs. So we assigned taxonomy as described before, and the only difference being that this time we used the PR2 database. And this is the best reference database for eukaryotic 18S that's available. And the great advantage is that it has standardized taxonomy. So this cumulative graph shows how well or to what rank we were able to annotate the OTUs. So on the leftmost, we have the domain, which is eukaryota on the left. And this goes down to species level on the right. So out of the almost 16,000 OTUs that we've processed so far, we could annotate the vast majority to the division level. And this is things like alveolata, rhizaria, straminopiles, etc. And we could annotate a good chunk to the order or family level, and then almost half of these to the species level with statistical support. So this is almost 9,000 OTUs labeled with the 18S and 28S sequences. And these annotated 18S and 28S sequences, they represent a huge increase in sequences available for several taxonomic groups when we compare it to the PR2 database. So for instance, our OTUs represent more Perkinsit sequences than present in the PR2 database. And we also see substantial increases for groups such as Cercozoa, for Discoba, for dinoflagellates, etc. And now here is an example of the kind of tree that we get. So this is a combined 18S and 28S tree of the deep marine samples. So these are the 5 mesopelagic and bathypelagic samples. This is a tree that's built only with our PacBio sequences, so there are no reference sequences here. And the branching level, or sorry, the branch colors represent to which level each sequence or OTU has been annotated. So the darker the color, the better, or the lower rank it has been annotated to. So the darkest ones here are the genus, and so on. So doing so, we can visualize, the diversity present in our samples, but we can also see how much of this diversity is novel. And this seems to be sort of correlated with how well the groups are represented in the PR2 database. For instance, the Syndeniales, which are really well represented in the PR2 database, they're mostly annotated to the genus level. On the other hand, we see novel diversity in groups like Diplonemids, and this seems to indicate that our OTUs have closed some gaps that are present in reference databases. So by doing this for all the environmental samples and by combining them, we should get a robust and comprehensive 18S, 28S eukaryotic tree. And I think this method can prove to be quite powerful with Illumina data, because by getting a more comprehensive or more complete tree, we can better place all of the Illumina data that exists, whether it's V4 or whether it's V9, it doesn't really matter. And by putting this Illumina data in the phylogenetic context, we can start to answer questions of an evolutionary nature on a really broad scale. So just to very briefly illustrate what I mean, one of the questions that I'm really interested in is how often protist groups transition across various environments. So are transitions really rare in general? And are some groups better at transitioning across environments than others? And do the transitions happen predominantly in one direction over the other? So these are the sort of questions that we can answer on a really broad scale now. And we can do this by placing the wealth of Illumina data that exists on our combined 18S, 28S tree, and then doing ancestral state reconstruction. So a scenario like this, where the different branch colors represent different environments, this would indicate a single transition event in this particular clade. Whereas a scenario like this would indicate multiple transition events. So to sum up, I've shown you that we can use long environmental sequences to build trees, and we can generate 18S, 28S databases. This can prove to be very powerful in combination with Illumina, and we should be able to use different Illumina data sets, even if they have sequenced different barcode regions. And with that, I'd like to finish by thanking a lot of people, all of the lab members, and in particular my supervisor, Fabian, our collaborators in Germany and in the UK, and all of the people who got us our samples for the environmental sequencing. Thank you.
Thank you, Mahwash. Do we have any question from the audience? Yes, I think Maggie.
Sorry, I was trying to make a clapping, not a hand raise.
Oh, okay. They look similar. Okay. Well, I guess I have maybe a naive question, but I mean, I guess the point of adding the 28S is to increase the resolution of your tree. But yeah, for your taxonomic affiliation, if I understood correctly, now you're going to be limited to the 18S, like to the resolution you can get with the 18S, right?
Yeah.
So
exactly.
Yeah. So what would be the way around that?
I mean, at the end of the day, our taxonomic annotation results, they will only be as good as the database in a way. And I think the 18S is the PR2 in particular, for example, it is quite a comprehensive database. So yeah, I'm not sure if I answered your question.
If I reword this, what kind of resolution, what kind of increase in resolution do you gain then by adding the 28S?
Well, it doesn't really increase the resolution for the taxonomic assignment, but just you get more robust trees at the end.
Right.
Yeah.
Right. Okay. Oh, there is a question from the audience, I think. Yeah, you can unmute yourself.
All right. Yeah. Well, very cool talk, by the way. I'm interested in, let's say, community structures and those kind of things. And I'm still struggling with the idea of PCR-based issues. I'm doing the same, so I'm not blaming you. But how do you deal with that? So you want to compare, I think, in the end, some kind of communities in the different systems. Have you compared that with, let's say, metagenomic approaches or some kind of PCR-free ones? And if so, or if not, what would you expect? And if so, what did you see?
Yeah, okay. Yeah, I mean, absolutely. That's a big problem. Maybe I can show you some. So I think my approach to this is that I'm trying to get, so we're not sequencing as deeply as we are with Illumina, I think. And so I wouldn't really use our PacBio results to draw any meaningful conclusions about community structure, perhaps. But of course, you can get some idea. But I would say the main aim is to generate longer sequences that can be used for placing Illumina reads in the phylogenetic framework.
Thanks.
Okay, I have a question from Mark Vandergiesen in the chat. So he's just asking if you've tried MinION instead of PacBio.
I was waiting for this question. I personally have not tried. I would be really interested to hear if anyone in the audience has tried it and what their results were. But the last time I checked, it's cheaper per high quality base to do PacBio than it is to do MinION. Yeah, I'm also really interested to hear how you get how you get around the high error rate with MinION, because at least with PacBio, and especially with Sequel 2, you're sequencing the molecules so many times that your error rate is actually really, really quite low as compared to MinION. Yeah.
All right. Any other question from the audience? Okay, well, thanks a lot, Mahwash.
Thank you.
Thanks. And our next speaker is Novák Vanclová. Anna, if you can share your screen. So in the meantime, yes, I will just briefly introduce Anna. So Anna did her PhD in Prague with Vladimir Hempel, and she's now started a postdoc in Paris with Richard Dorrell. She's an evolutionary protistologist, and her primary interest is the evolution of secondary plastid. So Anna, take it away.
Okay, thanks a lot, Laura, for the introduction. So my name is Anna, and I would like to tell you something about lateral gene transfer in euglenids and dinoflagellates. And this process is sometimes compared to shopping bags, where the organisms put various stuff and decide what to discard, replace, and keep on the go. So endosymbiotic events are major sources of lateral gene influx. However, the proteomes of complex plastids rarely contain only proteins of host and endosymbiotic origin. They are often chimeric. Plastid gains tend to coincide with LGT from other algal and prokaryotic sources, which can happen through one or more of the following general scenarios. So these would be A, the random transfer of the plastid-like genes, which then enables the plastid establishment. B, the concurrent gene transfer from both the endosymbiote and other algae, which speeds up the endosymbiote integration. And C, as a series of endosymbiosis and endosymbiotic gene transfers, each of which can readily recruit genes left by the previous one. Also, it is worth to mention that in primary plastids, there is a non-negligible presence of chlamydial genes. So some people theorize that the chlamydial endosymbiont may have been somehow involved in the primary plastid establishment too. So my previous work focused on euglenophytes. We analyzed mass spectrometry determined plastid proteome of Euglena gracilis, and among other things, we tried to sort the non-green genes into narrower categories. So up to 30% were of red origin, but only a tiny proportion of these were from primary red algae, while the ochrophytes and haptophytes, these two colors, are more likely donors of these genes. When we looked at the functions in the plastid, we did not observe any link between origin and function, except for slight enrichment of non-green genes among membrane transporters. However, there was a pretty interesting story waiting among us among the proteins of prokaryotic origin. There wasn't a lot of them, but we found there an additional full plastid localized subsystem for iron sulfur cluster assembly, which is of chlamydial origin, as well as several other sulfur metabolism related proteins. And this cannot be ruled out as a mere contamination or some evolutionary neutral transitional stage, because the full pathway was found in other euglenophyte transcriptomes too, but its function is yet to be determined. In my current work, I moved my focus to dinoflagellates, other group of complex algae. And one dinoflagellate group, which I'm now interested in, are Careniaceae, and they substituted their ancestral peridinian plastid by a new one from haptophyte. And it is even possible that the residual peridinian-like plastid is still kept in these. And my colleagues previously determined that the evolutionary rate of plastidial genes of Careniaceae is elevated, and that they possess a specific plastid targeting domains, which are different from the peridinian-like ones. And there is also one interesting recently described Careniacean, the Rossi dinoflagellate, which has kleptoplastids, which it obtains from a different haptophyte than the one which is the donor for Carinea and other type members. And these kleptoplastids are retained for unusually long time. So even with the relatively few studied species, the situation in Careneaceae looks already quite complex and interesting. For the preliminary prediction and evolutionary analysis of Carinea brevis plastid proteome, we can see that the plastidial pathways, which are usually kept even in non-photosynthetic plastids, such as core carbon metabolism and terpenoid and amino acid synthesis, are mostly of early or peridinian-like origin, while photosynthesis, pigment synthesis, and housekeeping genes of the organelle is mostly of late or haptophyte origin. But this requires more sophisticated phylogenetic pipeline and more data. So to summarize this, the contents of the proverbial shopping bags of complex algae can help us reconstruct their evolutionary and functional history. And for this project, we plan to generate new Careneaceae transcriptomes and attempt more comprehensive and large-scale analysis of their proteome composition and origin. And that's all from me. I would like to thank you for your attention.
Thanks a lot, Anna, and the beautiful slides. So once again, we don't have time for questions, but Anna will be in her own breakout room later if you want to talk to her. Thank you.
Okay, thanks. Now we move into the next larger or longer speak by Akinori Yabuki from the Japan Agency for Marine Earth Science and Technology, and we'll talk about the Cryptista. So if you can share your screen, that would be good.
Yeah. Can you see the slide?
Yep.
Okay. Thank you very much for giving me such a nice opportunity to introduce my work. So today I would like to talk about Microheliella maris. So this is the background information. So as you know already, there are various morphologically diverse organisms in eukaryotes, and there are many independent lineages in eukaryotes. Some phylogenetic relationship among them are understood well. However, other relationships still remain to be understood. For example, the relationships among Diaphoreticus members around them is not fully understood. So Cryptista, this is Cryptista, is one of the subgroup of Diaphoreticus. So Cryptista is composed of two subgroups, Rolomonadia and Palpitia. I think the early evolution of Cryptista is understood well compared to other eukaryotic subgroups. So only cryptomonas have the plastid of which origin of is red alga. Other Cryptista member does not have plastid. Recently, nuclear genome of a goniomonad was sequenced, and the information supporting the existence of plastid, including a remnant of plastid, was not reported. So therefore, I think it is now inclined to consider that the plastid of cryptomonas was acquired as the ancestor of cryptomonas around here. And just a moment. And this is ultra-structure. This upper picture is the TEM image of cryptomonas. So you can see thin electron dense layer here. This is paircraft. The cell membrane of cryptomonas and goniomonas was supported by this thin layer. So this is the characteristic of cryptomonas and goniomonas. And this characteristic has not been reported from Catabrephares and Palpitomonas. So the paircraft was acquired as the common ancestor of cryptomonas and goniomonas. On the other hand, cataboliophytes, the cell of cataboliophytes is covered by sheath structure like this. This is a cell membrane, and this area is sheath structure. This characteristic was only found in cataboliophytes, was not found in paractomonads and cryptomonads and goniomonads. So this characteristic was acquired at the ancestor of cataboliophytes, and paractomonads is naked. And cryptomonads, goniomonads, and cataboliophytes were collectively called as Loromonadia because these three subgroups has ejectosome in common. Ejectosome is a ribbon-like coiled ejective organelle. So this picture is showing the discharged ejectosome. Paractomonads does not have ejectosome, so this characteristic was acquired at the common ancestor of Loromonadia. Paractomonads is morphologically and ultrastructurally different from these, the member of Loromonadia. However, Paractomonads also has bipaptide flagella hairs. So you can see the flagella hairs, black arrows indicating the shaft, and white arrows indicate terminal hairs. Such bipaptide hairs are also found in cryptomonads. The shaft part of the shaft part of flagella hair of cryptomonads seems harder, much harder than that of paractomonads, but the structure is very similar to each other. And the goniomonad flagella hair is very simple. The F indicates the flagella hair of goniomonads, and goniomonads also has a flagella spine. That spine has not been found in other Cryptista. So flagella spines was acquired at goniomonads independently. So since paractomonads and cryptomonads has a flagella hair, similar flagella hair, this characteristic was acquired at the common ancestor, their common ancestor. So I want to say the early evolution of Cryptista is understood well compared to other organisms, other eukaryotic subgroups. On the other hand, the phylogenetic position of Cryptista is not stable. In this tree, Cryptista branched together with Archaeopteryx. Cryptista is a sister of Archaeopteryx. On the other hand, this tree, Cryptista branched within Archaeopteryx. Cryptista is here. Cryptista forms a clade with rhodophyta, and then rhodophyte and Cryptista clade is a sister of green and glaucophyte clade. So the phylogenetic position of Cryptista is not stable. These are background information of Cryptista. So then I would like to introduce today's reading part, Microheliella maris. I would like to call Microheliella maris MH in this talk because Microheliella maris is long to say. So MH was described in 2012 in this paper, in my paper. This is a light microscopic image of MH. You can see small cell with radiating some axopodium without flagellum. So MH is a typical heliogen-shaped protist at the light microscopic level. On the other hand, MH is counted as one of the orphan protists because MH has the unique ultrastructures. So you can see this structure. This is a MTOC. MTOC stands for microtubular organizing center. So many microtubes are radiating from this structure. The MTOC are the center of the cell. This arrangement is very similar to central heliozones, but also you can see the microtubes are penetrating nucleus. This is nucleus, and microtubes are penetrating nucleus. So this characteristic has not been reported from central heliozones. The penetrated nucleus was only reported from Heliomorpha. That is another orphan protist. So I want to say body plan is different between MH and central heliozoa. It's big difference. And further, so microtubular arrangement is also very important characteristic because microtubular arrangement is very conserved. And MH has a triad microtubular like this. So this arrangement was shared with Pedinelids. Pedinelids is a subgroup of foraminophyles, such as the famous Pedinelid is Ciliophrys and Pteridomonas. So they are foraminophyles, phylogenetically distinct from MH. And this is a combined ribosomal RNA gene tree. So MH is here. MH branched with Parapitomonas, but this position is not supported well. Therefore, I think, MH is a orphan protist, one of the orphan protists. In this study, we just conducted RNA-seq and DNA-seq analysis. So based on RNA-seq data, we did the phylogenetic analysis to know where MH goes. And we also conducted DNA-seq to decide the mitochondrial genome sequence of MH. Recently, mitochondrial genome of various protists has been sequenced, and its evolution is discussed very actively. So we just would like to do it with data of MH. So this is the result. So this tree was reconstructed by Yuki Yazaki, this smiling nice guy, at Riken, Japan, by analyzing 319 gene dataset. You can see MH is here. MH forms a clade with Cryptista with the highest bootstrap value. And MH and Cryptista clade forms is a sister of Archaeopteryx. And this is the result of a smaller dataset. MH is consistently shown to a sister of Cryptista here with highest bootstrap value. And MH and Cryptista clade is also sister of Archaeopteryx. And Archaeopteryx is shown as a monophyletic group. The spot is very weak, not strong, but Archaeopteryx is monophyly. So I think the phylogenetic signal of MH may contribute to grouping of Archaeopteryx because such a the size of analysis, the monophyly of Archaeopteryx was not reconstructed in such a set of analysis usually, but of course further data and analysis are needed about it. So this is the first my take-home message. Probably you are already in your home, but this is first take-home message. So we would like to propose a new clade name, Pan-Cryptista. So Pan-Cryptista composed of Cryptista and Endohelia, like this. This is a new finding. So as I explained, the early evolution of Cryptista is understood well. And now it was shown that Microheliella MH branched at the base of Cryptista like this. So what we can discuss based on this new finding, so I seriously think, but so unfortunately almost nothing because MH is morphologically and ultra-structurally very different from other Pan-Cryptista members. MH is just eccentric Pan-Cryptista member, so it's not flagellate helizoan shape. So the changing flagellate to helizoan shape just occurred around here, and their ancestors are possibly flagellates. So unfortunately, we cannot discuss in detail based on this new finding, but I'd like to propose a possible synapomorphy of Cryptista here. So blue arrows indicating blue arrows indicate a mitochondrion. So many members of Cryptista has a single mitochondrion like this. This is a cryptomonas. These are long weight shape of single mitochondrion. Such a similar arrangement of mitochondrion was also found in goniomonas and palpitomonas like this. Unfortunately, cadaveriferes have multiple mitochondria, but similar mitochondrial arrangement was also found in Microheliella like this. So I'd like to suggest this cytoskeletal mitochondrion may be a synapomorphy of Cryptista. However, many members of Cryptista are just small cells. Therefore, it may be just due to the small cells. So, but some cryptomonas have large cells, but they have a single mitochondrion. So I want to think this characteristic is a synapomorphy of Cryptista. Okay, so I'd like to move to another subject about mitochondrial genome. But this beautiful image is summarizing the structural diversity of protist mitochondrial genome. Some protist has circular chromosome, and another have linear chromosome, and the other ones have multiple linear or multiple unicircular chromosomes. So there is a big structural diversity in mitochondrial genome of protists. And their gene content is also different. The gene content on the mitochondrial genome is different lineage by lineage, and its reductive evolution is also one of the hot research subject recently. And this year, really nice paper was published from Nature Microbiology. And in this paper, so many new mitochondrial genomes was reported, and update in this paper, the possible ancestral state of the mitochondrial genome of last common ancestor of Diaphoreticus was introduced. So Cryptista is known to have the gene richest mitochondrial genome of Diaphoreticus to data. Cryptista has 47, and the mitochondrial genome of Cryptista has 47 proteins, which is just 8 less than Diaphoreticus ancestor. In other words, the mitochondrial genome of Diaphoreticus has 55 proteins. And this is the result of mitochondrial genome of MH. We successfully decided a full genome sequence of mitochondrial genome of MH. This is a 61 kilobase pair of circular chromosome. And there is a pair of inverted repeat red arrow ones are indicating inverted repeat. So on this genome, there is 3 ribosomal RNA genes, 1 RNSP genes, 21 transfer RNA genes, 1 transfer messenger RNA genes, and 53 protein coding genes. 53. So it's very surprising result to me because it was suggested that the common ancestor of Diaphoreticus had 55 proteins. So this is the updated gene list of various protists. So this second bottom one is showing the gene content of micro-MH and the updated gene content of last common ancestor of the diaphoreticus is here. So red one is a newly identified genes. So this is TAF-A and this one is COX-11. TAF-A and COX-11 has not been reported from diaphoreticus members before. So this is the first report. And this is a phylogenetic tree of TAF-A and COX-11. So you can see my this is TAF-A tree and this is COX-11 tree. You can see TAF-A branched here, branched within empty mitochondrial genome encoded crate like this, and MH is here. So I want to say just based on this result, TAF-A and COX-11 of MH have been vertically inherited from the common ancestor of eukaryotes because they form both sequence branched within the crate of eukaryotes. But this is this image showing the summarizing the evolution of mitochondrial genome. The last common ancestor of eukaryotes has the mitochondrial genome of last common ancestor of eukaryotes probably have 69 proteins, and the last common ancestor of diaphoreticus has 57 proteins on its mitochondrial genome. And this point A to B, which is a common ancestor of Archaeopterygidae and Cryptista. From A to B, just two genes, RPL18 and RPL27, move to nuclear genome from mitochondrial genome. And from B to C, which is the common ancestor of Archaeopterygidae, another 7 genes move to mitochondrial genome to the nucleus. And from B to from B to D, another 8 genes move to nucleus genome, from mitochondrial genome. And from B to E, which is Microheliella maris at the present day, just 2 genes move to nucleus genome to nucleus genome from mitochondrial genome, just 2 genes. So of course, I don't know why Microheliella is keen to keep many genes in its mitochondrial genome, but this information is very useful to understand the ancestral state of the mitochondrial genome of diaphoreticus. So only 4 genes move to nucleus genome from mitochondrial genome in Microheliella. So this is a summary of this study. MH branches at the base of Cryptista. In the Microheliella and Cryptista are should be called as Pancryptista collectively. Cytoskeletal mitochondrion may be shared in Pancryptista, and I think this is a possible synapomorphy. And MH, Microheliella, has the most primitive empty genome of diaphoreticus. So thank you for your attention. This study is almost all phylogenetic tree was reconstructed by Yuki, and Yuki and Yuji Inagaki are the co-PI of this study. And Takashi Shiratori also helped to sequence the mitochondrial genome of MH. So that's it. Thank you very much.
Thank you, Akinori. Very, very nice overview and work. Does anyone in the audience have any questions? Just wait for raised hands. Just a question. So the Yakubids are generally known to have the largest mitochondrial genome composition. So did you do a comparison between them? So you have nothing special that is not present in Reclinomonas or in the Jacobids in general?
Yeah, Jacobids has, as you know, Jacobids has more genes, but so There's, I'll just say, so yeah, there is no special things to compare about between Jacobi's and Microheliella. So
No, okay. So your Microheliella is just a real subset of the Jacobids. You have no additional genes.
no.
No. I think in your original paper for Microheliella, I think I might be wrong here, that you described it as a Heliozoan.
No, so
the 2012 paper, I thought, but I might have misremember.
in this paper, so Microheliella was classified into different groups from Central Heliozoa. So Microheliella was classified into Endohelia, which is different from Centrohelia.
Okay, yeah. I'm not sure if Philip has a question or if he's...
Yes, sorry, as a host, I cannot raise the hand electronically. So my question is, in your multi-gene phylogeny with monophyletic Archaeplastida, does that include Rhodelphis?
We included, so yeah, probably that protist also contribute to recover the monophyly of Archaeplastida.
But it goes sister to red algae. It still goes sister to red algae.
Yeah.
See. Thank you.
Good. Anyone else?
Sorry,
not,
I cannot raise my hand for some reason. I sent you a question in the chat, and that I don't know if you saw it, and that was if there is any comment in this organism of the presence of introns, class 1 introns or class 2 introns in the mitochondrial genome.
So no, we couldn't find any intron in the
Any introns at all?
No.
Okay. Thank you.
Okay, thank you for the question. Now, if anyone else has questions later on, then we also still have the final breakout room after the next speaker where people can talk to you as well. Okay, thank you, Akinori. Then we move on to the next speaker, Tristan Biard. So Tristan will talk about some Protist and ecology, and he's at the Laboratoire of Oceanology and Geoscience in France. Tristan, if you can share your screen.
Yeah, just a second, please.
Oh, yeah. You're still there, Tristan. It looks like you disappeared.
I don't think he's connected anymore.
No, I didn't see him on the list anymore.
Maybe we can share the slide, Andres, with the meet.
Oh, there he is.
IDs.
He's back.
Can you hear me?
Yes, you're back. Good.
Yeah, sorry, there was a problem with sharing screen issue. Okay, do you see my screen?
Sure, yeah, go ahead.
Okay, perfect. So thank you for the invitation. And during this talk, I will try to answer a simple question. Can Protist and ecology be inferred from in situ images? So yes, I will not waste some time. So yes, we can do ecology with in situ images. And I will try to explain how we do it and what are the limitations. So I think John is not here, so we will forgive me about sharing this photo. So you can do ecology the old-fashioned way. It's usually very reliable. And how do you do it? You just use buckets, net, Niskin bottles. But the thing is that we all know that with these tools, even the most expensive one, you will have a poor vertical resolution, meaning that you cannot really go see and do single cell ecology. And also, if you want to relate the distribution of your cells with environmental variable, temperature, salinity, this will be an indirect way. You can put CTDs in the water, but it won't be at the exact same place where you collect your cells. So you could do culture too, but most of us know that our protists of interest are not in culture. So you have to deal with that. You have to go and fish them in the ocean. So how can you do do another way? You can simply go and see where they are. So here you have an illustration of bugs in the ocean, and you can see jellyfish that are not protists, obviously. But if you look more closely into this image, you can detect some single cell organism. So you see the scale on the top of the image is 10 millimeters. So obviously everything here is on scale, and the protists that I'm showing are very, very big. I'm not mentioning the 2-micron stuff that you can see in the ocean. So this is a limit, and this will be the limit for my talk of today. So these protists are rhizarians, radiolarians and phyllodarians, if you want more details, and I will focus on them. So how can you obtain such images? So first you need an in situ camera. So here it's under vision profile of 5. It's the 2 red tubes that you have at the bottom of the image. And what do you have on this rosette? So if you went on ships, you know that on rosette you can have Niskin bottles, and from Niskin bottles you can measure dissolved nutrient. So not at a very fine vertical scale, but at least you can get some numbers, and then you can do some interpolations to have numbers at finer scales. Then along with the camera, you have CTD, and CTD can get you temperature and salinity at very fine vertical resolution. You can also have oxygen sensors. You can have information about particle numbers, about nitrate concentration, and also you can get some PARs if you have a PAR sensor at the top of the rosette. Meaning that if you get these pictures from your camera, so here it's a phyllodarian called Holosphaeridae. So what do we know about this cell? First, we know its size. So it's about 4 by 4 millimeter. We know that this cell was taken and observed at 287 meter, plus or minus 20 centimeter resolution. So it's a very, very fine scale resolution. And we know that this cell was observed at 8.2655 Celsius, which is very accurate. We can get the same things for salinity, oxygen. The PAR and the fluorescence was obviously close to 0 given the depth. We can more or less know the concentration of silicates based on interpolation from discrete measurement taken with Niskin bottles. And we can get particle concentration for different size classes. So here I just take the 1 millimeter limit, but this is just a random cut. And we can get the same numbers for basically, in my data set, 22,000 images of individual rhizarians. And what do you do from this? Well, basically you do statistics. And you can use additive models to look how well you can predict the abundances based on environmental variables. So you have 3 different panels, which correspond to 3 layers in the ocean. You are on the left, you have the upper epipelagic, which could be considered as the euphoric zone where you have light. Then you have the lower epipelagic, where there is more or less no lights. And then you have the upper mesopelagic on the right. And from the R square, you can see that the prediction is more or less good, depending on where you are. But still, it's pretty accurate, especially for the lower epipelagic region. So we can predict. But now what we want to know is, if we have these individual cells, can we go into finer details? If I take the example of this bug, so you have an illustration on the top left, and this is a Phylogenian, and we can go and look at, in different stations, the accuracy of prediction using different environmental parameters. And here we detected that dissolved silicon or silicates concentration and temperature had some significant effect on the distribution of this bug. So you could say, okay, well, this is based on very localized sampling stations that were made in California currents. So can we do this at a global scale? The answer is yes, we could, because we could use some of the very large collection of in situ images that are hosted in Ecotaxa. So you have the link down below. And on this link, on this website, we have something like 3,000 vertical profiles. And in this 3,000 profile, we have more than 100,000 individual images of rhizarian that are distributed in different places of the ocean. And now if we are using more complex models and with indirect environmental variables that are taken from the World Ocean Atlas, we can end up having some such maps where you can predict biomass from individual images, which is pretty exciting. So the answer of this question is yes. That's what I said at the beginning. And here on this very short talk, I only focus on a very big fraction, which is about something larger than 1 millimeter, which is obviously a very small fraction of the Protist. But we have more and more devices that are coming on the market that are more focused on smaller fraction. And we could do the same thing with other Protists. And this is a very exciting future for us working with in situ imaging. So on that, I would like to thank the organizer and leave you on this weird picture of a colonial radiolarian that is being used as a surfboard by some kind of mizzen. Thank you very much.
Thank you, Tristan.